Friday yes. Dear student, today we will discuss about cell division, in which second part of cell division is a meiosis. Meiosis is also called as reduction division. Why is called as reduction division? Because the chromosome numbers are reduced to half. So here, meiosis is the type of cell division by which germ cell, egg, and sperm are produced. It is a specialized and rather complicated type of cell division occurring only in the diploid reproductive cell and result in the formation of haploid sex cell or gamete. Daughter cells have half number of the chromosome found in the original parent cell, as first of all described by Weizmann in 1887. As you all know, there are two types of meiosis: meiosis first and meiosis second. Meiosis first is called as the heterotypic division, and meiosis second is called as the homeotypic division. So let's see here. There is a meiosis first, that is a heterotypic division. It is called as a heterotypic division because of it there is a process here has a longest there is a duration, and it is having the 5 substages like leptotin, zygotin, packaging, diplotin, dikinesis, and after completion of dikinesis, again there is metaphase first, then anaphase first, then telophase first. So see here what is happening in the leptotin, zygotin, packaging, diplotin, dikinesis, and metaphase. So this is the uh, leptotin is a first substage of prophase. Here, what is happening? The volume of the nucleus is a increased, considerably increased. Here, which you are in the picture, you are see. Yeah, the chromosome become distinct, long and uncoiled. They occur in a diploid number. They contain a series of bead-like appearance on chromomeres, and the centriole divides into two. So such type of there is a chromosomal changes is there is happening in the leptotin. Then it convert into the zygotin. Two homologous chromosomes approach each other and began to pair. Here pairing is going on. This pairing is called as a synapsis. Here synaptomial complex, which is shown in the diagram. Each pair consists of maternal chromosome, the chromosome from mother, and paternal chromosome from there is a another is a father. So this is the one is red, and another is a blue color is there is shown very clearly, and so that they are joined to form a bivalent. Bivalent, bi means two, valent means there is a bundle. One bundle is paired, and later on what is happening? The chromosome becomes shorter and thicker. Here, shortening and thickening of the chromosome is happening. The nucleus increases in size, and the centriole moves to the opposite pole. This centriole here, there is a move to the opposite pole. Here there is a it is shown here in the diagnosis. There is a it is a start from there is a this zygotin stage, but here it is not shown in the diagram. The process is there is a going on from this stage, and later on there is a next stage is convert. There is a packaging stage. Each individual chromosome of each bivalent began to split longitudinally into two similar chromatids. As a result, each bivalent now contains four chromatids. This is called as a tetrad stage. So here, tetrad formation is there is a takes place. Bivalent is converted into tetrad. Tetrad means there is a four. So here there is a four centromeres of this is a split into two, and there is a tetrad formation is takes place, and there is a the homologous chromosome of each pair begins to separate because of the gradual disappearance of the force of attraction between them. However, two homologous chromosomes do not completely separate but remain attached together at one or more points as indicated by X arrangement. So here they are there is a separating with each other, and after this end of this is the uh, completion of a packaging, and this later on the diplostene is there is a happening. And what is happening in the diplostene? The homologous chromosome repel each other. Two homologous chromosomes thus separate from each other, and there is a somewhat they are having attached to the there is a chromatid. There is a happening, and there is a such type of a the crossing which is take place at the terminal position of there is a chromatid, that is called as a chiasmata, and this later on convert into dikinesis, which is the last stage of there is a prophase first of meiosis, that is called as a dikinesis. in which what is happening, the chiasmata is going to separate it. That process is called as a terminalization. So terminalization is there is a takes place in the dikinesis. They are going to separate. The valvulae tend to repel each other and migrate to the periphery of the nucleus, just inside the nuclear membrane. The nucleolus disappears here, which is it is a completely disappearing. The nuclear membrane also begins to disappear. Here, this nuclear membrane also having disappearing. The spindle fibers make their appearance in the cytoplasm, and there is a spindle fiber is there is initiated, and there is a metaphase. First is there is a takes place. There is a after prophase first there is a metaphase there is a happening, and here in the metaphase, in the mitosis of metaphase and meiosis of metaphase, what is happening at the very first? What is happening? Formation of the spindle fibers. Here, spindle fibers is there is a initiated from there is a opposite there is a centrioles, and there is the chromosomes which are separated from there is in the dikinesis. They are having came into the a central line in the equatorial line. So this is shown in the diagram in the last here metaphase in the picture here. So what is happening? There is a the again the chromosome of these two. There is a metaphase of mitosis and metaphase of meiosis having again somewhat different. Both are having in the central line, but in such a manner that their centromeres are toward the center in the mitosis, and here there is in the metaphase of the center, the arms are toward the center. So this is the somewhat difference between there is a metaphase of mitosis and metaphase of meiosis. So this is clearly seen in the diagram. So in this way, there is a after metaphase, there is a next one is anaphase. So what is happening in the anaphase? The opposite, the chromosomes are goes to there is a up the force of attraction between there is a the toward which are came into the center, they lapse and they move to the opposite direction, or there is a spindle fibers drag the a respective chromosome to each pole. So there is a in the anaphase chromosome pair separate with sister chromatic and remaining together. So means there is a the shortening of the spindle fibers and there is a chromosomes to the opposite direction. Then the last is the telophase. Two daughter cells are formed with each daughter containing only one chromosome of the chromosome pair. So here in this way two cells are formed. Here there is a metaphase. It looks like this manner, and here this is anaphase. They are opposite in direction. These are spindle fibers which are having there is a shortening of the spindle fibers or there is a takes place, and here they move to the opposite direction. The homologous chromosomes move to the opposite pole of the cell. That is a anaphase. This one here. Equatorial plates. These are there is a in the form of a chromosome or equatorial in the opposite direction. Arms are toward the center. So these are the diagrams of metaphase, anaphase. First. This is the first, and this is the telophase first. So what is happening? There is a chromosome gather into a nucleus and original cell divide. So this is a single divide, cell is divided. First of all, there is a cell wall is formed in the center and it gradually moves to the periphery and two cells are there is a formed. And after two cells are formed, the chromosomes which are to the opposite poles, they are there is a lose there is a their identity and they become reticulate nature, and there is a again new nuclear wall is developed to the both sides. Nucleus is again reappear, and in this way there is a having a, all the content of there is a parent cell are they are synthesized and two exactly similar cells are there is formed in the two a telophase first. So two daughter cells are formed. Then there is a meiosis is again goes into the meiosis second, and this is called as a homeotypic division. That first is a heterotypic. Why this is a heterotypic? Because the prophase first is having a longest duration and having 5 substages: leptotin, zygotin, pectin, diplotin, and dikinesis. So that this is a Hetero means different, unequal. Homo means similar. Meiosis second is a homeotypic division. Similar divisions are there. And meiosis second is exactly similar to like that of a mitosis. Whatever changes are in the chromosomes and there is a in the daughter cell, that is a similar in the meiosis second. So what is happening in the prophase second? Prophase 2, metaphase 2, anaphase 2, telophase 2. These are there is a sequence of there is a phases of there is a chromosome. There is a cell which is again there is a similar like mitosis, meiosis first and meiosis second. So what is happening in prophase second? DNA does not replicate, and in the metaphase second chromosome line up at the center of the cell. Metaphase is there is again a same there is a characteristic feature of there is a chromosome. All chromosomes are gathered to the center, to the equator, or to there is a they line up at the center. That is a metaphase. Whenever you see there is a chromosomes at the center, you there is a confidently called it is a metaphase. Then anaphase. Anaphase, there is a chromosome replicate to there is a opposite direction. Means they are moves to the opposite direction. Centromere divide and sister chromatids move separately to each other. They migrate toward the there is a opposite direction. So that is a characteristic feature of anaphase. And there is a telophase is a cell divides and it is cell division is completed. Means there is after there is a cell wall is formed the after anaphase, and there is a two daughter cells are there is a form. Means here there is a in after telophase there is a four cells are formed. यहाँ पे क्या होता है दो दो सेल में there is a होता है. So there is a prophase two, phase, metaphase two, anaphase two, telophase T is there is happening. Four haploid daughter cells are formed after there is a telophase second of meiosis, and this is a meiosis. There is a second one. So after meiosis, there is a how many cells are formed? Four cells are formed. After mitosis, two cells are formed there. Here, this is the difference. So see here, this is the diagram. There is a after telophase first, there is a two daughter cells are formed, and these two daughter cells later on again convert into four cells. So this here, this is professor. Two cells are there. They are there is a having DNA does not replicate. Just we have seen. Then in metaphase second, what is happening? The chromosomes line up toward the center, here the equatorial line. In anaphase second, they move to opposite direction, pulled opposite to the opposite poles, means opposite direction. See here, these are the two cells and these are the chromosomes to the opposite pole. This is a one pole, another pole. This is one pole, this is a, so these are the going to another pole. So genetic exchange of the material takes place in the packaging stage. Now the packaging stage में exchange हुए वाला color there is a which is shown here. So this is a diagram. Then telophase second is there. There is a two cells are converted into four cells are there is a the chromosome gather into nuclei and the cell divides, and there is a each of four cell has a nucleus with haploid number of a chromosome. Here there is a the parent cell is having there is a diploid number of chromosome, and this daughter cells are having there is a haploid number of a chromosomes are there is a form. So this is after meiosis, four cells are formed, and these four cells are not similar to its parent, and they are having only in the quantity, not quality. And here there is a chromosome number is reduced, so that this is the meiosis is sometimes called as a reduction division because of a the chromosome numbers are reduced to half, so that this is called as a reduction divergence. See here some diametric representation, that is how the one single cell is divided into four cells. 1, 2, 3, 4 here. The last diagram is 4. So here interface, 2 n number of a chromosome here, homologous chromosome is there is happening. After meiosis, first two daughter cells are formed, and these two daughter cells later on again converted into there is another two daughter cells, and there is a see here there is a meiosis second is there is happening in the third position, and this is a n number of daughter cells are there. Here parent cell is having 2n number of chromosome and daughter cells are having there is a n number of chromosomes. So in this way there is a chromosome is reduced to there is a half. So this is a called as a reduction division. So see here just some basic difference of mitosis and meiosis. Just we have discussed mitosis previously. and here just now there is a meiosis. So what is happening in the mitosis and meiosis? Mitosis is a somatic cell division, and it is a occurs in the reproductive cell. So meiosis is having a takes place in the reproductive cell, and it is happening in the vegetative cell, somatic cell, and this is a first difference. Then cell divides once and here cell divides twice. ये क्या होता है कि देर इज अ मियोसिस, दो बार देर इज डिवाइड होता है, दो बार दो डिविजन होते हैं, मियोसिस फर्स्ट मियोसिस सेकंड. Mitosis not having there is a such type of a two division, only single division is there is happening and two daughter cells are formed. And number third one is the two daughter cells formed, genetic information is identical. Here there is a qualitative and quantitative distribution of genetic material is occurred in the mitosis, whereas in the meiosis, not qualitative and quantitative distribution of genetic material takes place, and there is a daughter cells are having there is a half number of a chromosome, and that is a reduced number as compared to its parent, and so that there is a the meiosis is called as a reductional division. So this is a another difference of this mitosis and meiosis. Then next is the chromosome number is maintained. Here there is a the generation after generation chromosome is number is maintained. Means there is a same number is there is a happening in the daughter cells. Here the chromosome number is reduced. Here the chromosome number is reduced in the meiosis. This daughter cells are having a half number of chromosome, and it is a simple division. Mitosis is a which called as a very simple divisions are there is happening. It is a meiosis is a complex division. Why it is complex? Because of a the process of uh, meiosis first is a longest duration, which having five sub stages: leptotene, zygotene, pachytene, diplotin, dikinesis, and later on metaphase first. So that this is a very much complex and complicated division is there is happening, and there is a four cells are formed, so that it is called as a complex one, and this is very much simple. No, there is a such sub stages are there is a happening, like there is a process of meiosis first, and here mitosis process is very simple. There is a nucleus and nucleolar membrane disappear and chromosome set free into the wandering here and there in the cytoplasm, and later on they come to the a center line. So such type of a process is there is a takes place. Again, much more difference is happening in the mitosis and meiosis of every stage. Process of there is a mitosis is having again there is a changes. Meiosis first is there is having changes. Metaphase of there is a mitosis is again change. Metaphase of meiosis again changes in the chromosome configuration or chromosomal there is a location or shape or there is a all these things are different. Anaphase of mitosis is again different. Anaphase of meiosis also there is a different. Telophase is also different. Two daughter cells are here. Telophase is again different. Four cells are formed, but there is a variation in the maternal and paternal chromosomes are combined. There is a join to form a there is a new genetic there is a chromosomes are there is one. There is a the variation in the chromosome is there is takes place. So this is the you can see there is in the books of cytology which I have told you previously that there is a having a much more difference happening. Then last one is there is a significance of meiosis. So meiosis is a logical necessary part in the life cycle of sexually reproducing animals, since it leads to the formation of a gamete. So it is a very much necessary part. Every zygote is they having a meiosis, and so that there is a daughter cells are having there is a male and female gamete unite to form a zygote. There is so the n number of male gamete and n number of female gamete they unite to form a 2n number. So such type of a logical part is there is a happening, and it is a necessary part in every there is a reproducing sexually reproducing organ, and so that there is a formation of a gametes takes place. Then next there is significance of meiosis is due to meiosis transformation of diploid nucleus into haploid nucleus. Just I have seen we have seen here if there is a reduction division is happening, diploid nucleus is converted into haploid nucleus. So this is a you have to write one of the significance. Then Next one is the meiosis leads to the variation which provide the raw material for evolution. So here, in the packaging stage of prophase, what is happening? There is a chromosome crossing over takes place in paternal and maternal chromosome, and so that there is a the variation in the chromosome is there is a happening. So this is a raw material for evolution. New species may arise. So this is another significance. Then next one is a constant number of chromosome is in a given species is maintained. So every there is a species having there is a same number of a chromosome. That means there is a supposed to be band 46 chromosomes. So generation of human beings are having 46 chromosomes are there. So human beings are there is a all over the world having there is the same number of chromosomes 46, and then monkey is 48, 49, then onion, potato, all these species are having different number of chromosome numbers are there. So that is maintained due to the meiosis. Meiosis takes place so that it is maintained generation after generation. Then as the result of meiosis, four cells are formed. So after meiosis is happening, how much cells are formed? There is a four cells are formed. So this is there is a takes place in this there is a meiosis. So the this question maybe I also asked for full mark. There is a what is meiosis? Describe the significance of meiosis. And this is or what is meiosis? Describe process first of meiosis. There is a leptotin, zygotin, packaging, diplotin, and diaconesis. So I have shared you a record book of there is a our paper cytology and genetics. So in which I have given all details, diagrams, and there is a description of mitosis and meiosis. And this is also a compulsory question in the practical also, which having there is a 10 marks, and there is a you have to show a temporary prepare a temporary slide of meiosis and show a one stage to the examiner with well labeled diagram. So this is a compulsory question. Question number 2. Question number 1 is mitosis. Question number 2 is meiosis. And this is asked for a 10 marks in practical examination. So you have to prepare it for theory as well as practical. So you have to prepare a one time. It is used you in there is a two different there is a in examinations, practical examination and theory examination. And so that it is very much simple thing which is you have studied previously in previous there is a classes, and you have to read carefully, you can understand very okay. So I think today stop here, we will continue tomorrow.